of course, have the pleasure and honor to introduce Rainforest Trust's newest board member, the celebrated science and conservation author, David Quammen. David's published 17 books and numerous articles for, among others, National Geographic, The New Yorker, and more, winning prizes and fascinating people in topics spreading from island biogeography and extinction to the spillover of viruses from animals to people. His latest book, Breathless, which we're excited to announce comes out today, is about the science of SARS-CoV-2. He got a great review, among others, from the New York Times this past weekend. So we're really excited to have you on our board, David, and really grateful that you're willing to spend an hour talking to us about all these topics.
Thank you, James. I am very glad to be on the board, very grateful to have been invited and welcome to it, and happy to talk to you all today.
Great. So first of all, welcome to Rainforest Trust, and why on earth did you agree to join our board?
Because your mission is so important and your reputation is so stellar.
Nice.
Simpler.
Very nice. Thank you. Thank you. Well, tell us a little bit about your latest book, Breathless.
This is a book about the virus itself, SARS-CoV-2, the COVID-19 virus, its origins, evolution, fierce journey through the human population, and about the scientists who study it. And it's titled Breathless, partly because of the respiratory aspect, but also partly because it has been a breathless chase the last 2.5 years for science to try and characterize this virus, scrutinize its genome, figure out where it likely came from, how it likely functions, learn about it on the fly, learn enough to begin another breathless effort at developing vaccines and antivirals to deal with it. So this is a book about molecular evolutionary virology, which may sound deep in the weeds, but this is also a book about people. This is a book about heroic and brilliant scientists around the world, some of them very famous, some of them completely unknown to the public, who have done amazing, arduous work on this virus to allow us to cope with it even as well as we now are coping with it, which of course is not well enough.
Well, talking about the breathless speed with which the world had to deal with this, including the geneticists who live in your book, how some people remain skeptical of the mRNA vaccines in part because they were produced so incredibly quickly compared to any vaccines in history. How was it that we as a scientific community and political community, in spite of all the other problems, were able to produce vaccines so fast?
The mRNA vaccines were able to happen so quickly because some scientists had a running start of years working on the concept of an mRNA vaccine, and in particular 2 different teams, one of which led to the Pfizer vaccine, the other of which led to the Moderna vaccine. In my book, I focus a bit, you can't tell the stories of all the threads that have led to all the vaccines, so I've picked one to focus on, and that's the Moderna vaccine. Moderna is the name of the private company that produced it. The science came to a great degree out of the Vaccine Research Center, which is part of the National Institute of Allergies and Infectious Diseases, which is part of the National Institutes of Health. Therefore, Tony Fauci is the boss of the Vaccine Research Center. And there is a fellow there, a senior researcher named Dr. Barney Graham, who I happen to know from 10 years ago or so. And he emerged as the leader of the effort on the mRNA vaccine within the Vaccine Research Center. And that was logical because Barney Graham has been working on the concept of mRNA vaccines for years himself, working first toward a vaccine on well, I'm forgetting the technical name of it right now, but and that's partly because I'm a little bit befogged with my own case of COVID right now.
Right. I should thank you for mentioning. Thank you so much for joining us. You said you're on the rebound week from Paxlovid, the antiviral drug.
Right. Yeah. And I had over the last 24 hours, I felt like a fellow with a very bad hay fever attack who also had earache. But
thank you for joining us anyway.
Very, very lucky and mild compared to what so many other people have gone through. Anyway, I think it was vesicular stomatitis vaccine, a virus that infects children all over the world, VSV, that Barney Graham was working on for years and moving toward an mRNA vaccine concept, and then worked for a while developing an mRNA vaccine for the MERS virus, the coronavirus that emerged from Saudi Arabia, reservoir host, probably camels, or at least an intermediate host camels, a very lethal, dangerous virus, but not tremendously transmissible human to human. So it hasn't caused a pandemic, but it's caused a lot of concern. So Barney was up to speed with a number of other younger colleagues, including a fellow named Jason McClellan, who is a structural biologist who was very interested in this, and he'd gotten to know Barney Graham. So they were working on this for years. Let's develop a MERS vaccine using the mRNA technology. And then this thing arose, and I tell this story in the book. Jason McClellan, he's a young guy with a couple of kids, and he's in Park City, Utah on a Christmas vacation, getting a pair of snowboarding boots heat fitted to his feet, and his cell phone rings, and he looks and he says, "Oh, it's Barney Graham. He's probably calling to wish me happy holidays." And he answers, and Barney says, "Jason, this thing in Wuhan, it's a coronavirus. We got to do a vaccine. Are you in?" And Jason McClellan said, "Of course I'm in, Barney." And so they started. But what they needed to begin was, first of all, the genomic sequence, the 30,000 letters translated from RNA, because SARS-CoV-2 is an RNA genome virus, but RNA is very unstable. So when you sequence it, you convert it into DNA, and then you sequence the DNA. So there was this 30,000 letter genome of DNA that was the blueprint of this virus. And Barney and others were breathlessly waiting for that. I asked Tony Fauci, as I asked a number of my scientific sources, what's the most important decision you made in 2020, in the year 2020? And Tony Fauci said, well, two decisions, one political and one scientific. Political, speaking out against the president. I take no great pleasure in speaking out against the president. And then he said, scientific decision, well, that would be getting the genome as soon as we could and getting it to my people at the Vaccine Research Center, getting it to Barney Graham and saying, okay, here's the sequence, go. I will get you the money. I will get you the support. Just start right now, this hour, developing an mRNA vaccine for this virus. And that happened in the evening of January 10th, 2020. The sequence came out on a website in Edinburgh, having been sent there from a scientist in Sydney, Australia, who was working with scientists in China. He got permission from the scientists in China to reveal the genome. He sent it to his friend who runs the website in Edinburgh. Edinburgh posted it. Barney Graham is sitting in Washington or Bethesda. He gets it at 8 or 9 p.m. on January 10th and immediately passes it along to his young people in his lab. And they start that weekend. That was a Friday evening. They start that weekend working on the structural principles necessary for designing the antigen, the signal to your immune system that would be injected into all of us in the form of messenger RNA. So that's one of the sequences events that rather breathless.
So various people were prescient, but you were also prescient. Your previous book, Spillover, seemed to predict this pandemic. Was the pandemic predictable, or did you just get lucky?
I paid attention to the scientists who saw this coming, saw aspects of it coming. And there were aspects of it that were very, very predictable, heartbreakingly predictable. There is a scientist named Donald S. Burke. He's emeritus dean of the University of Pittsburgh School of Public Health. And he had lectured and written about the evolvability of viruses in the late 1990s. And I had spotted what he had been saying about this. So I interviewed him when I was doing my book Spillover. And I said to him, as I said to some other people, this was back in about 2010 or 2011. So is there a next big one coming, a next big pandemic? And if so, what does it look like? And he said, yes, there is a pandemic coming. We can't say when, but we can say with a fair amount of confidence that it will be caused by a virus coming out of a wild animal, an RNA virus, a single-stranded RNA virus, because those mutate readily and therefore they evolve quickly and they can shift from one kind of host to another. It'll come out of that wild animal somewhere where humans come in contact with wild animals. For instance, a wet market, for instance, in China. So he told me all that, and I put all that in the book in 2012. I wasn't prescient. I was just barely smart enough to pay attention to these very smart people who could see this coming. But he couldn't say when. Oh, and he also said it will probably be either an influenza virus or a coronavirus. So that much we had. Anybody in this field, in that field, because I'm not a scientist, in that field, anybody who was a disease scientist, a molecular evolutionary biologist could have told you that, yeah, there will be a pandemic and it'll be caused by a single-stranded RNA virus because they are so highly evolvable.
Wow. Wow. If you don't have to answer this next question because maybe people need to read the book to answer it. So it's up to you whether you answer it or not. But did COVID spill over from wild animals to humans by accident? Or was it released by mistake or on purpose from a lab in China?
Okay, so I'm happy to talk about that, which is different from answering that question. I'm happy to talk about it. I think of those two as the natural origins school of thought versus the nefarious origins school of thought. And the nefarious origins school of thought encompasses accidental lab leak of a manipulated virus, engineered virus, all of those things. The evidence is overwhelmingly on the side of natural origins, overwhelmingly. But is the question answered with finality? No, not yet. Do we need to do more research on both sides of those two schools of thought? Yes. In particular, we need to do much more work on the natural origins scenario in the form of sampling viruses from many, many more bats and other wild animals in China and Southeast Asia in order to look for and try and find the progenitor virus of this virus, the virus that is 99.5, 99.8 similar in its genome to this virus. Scientists have viruses that are 96.2% similar in genome to this virus. Another one that's 96.8 or something like that. One from southern China, one from Thailand. Very similar viruses, but to say a virus is an RNA virus is 96% similar in its 30,000 letters of genomic code is to say that it's a cousin that diverged maybe 40 or 50 years ago. So it's not the virus that became this virus, but it's possible with enough field work, with enough sampling, that a much closer match can be found, and that effort needs to be made. But it takes time. Some people say, well, it's suspicious that they haven't found the progenitor virus in an animal yet. No, it's not suspicious. If you know anything about this field, you know that it takes time. It took 14 years to confirm the reservoir host of the original SARS virus. It took 41 years of effort to confirm the reservoir host of the Marburg virus. Marburg is related to Ebola, very dangerous virus. It takes time, or it frequently takes time. So we've only begun the effort to look for the natural origins. And in terms of the other school of thought, Um, I take the arguments from that school of thought very seriously and I hope respectfully in the book. I don't find empirical support for them. It's difficult or impossible to prove a negative. So it can ever be proven that this virus did not leak from a lab? Well, maybe it can't to the satisfaction of everybody, but there's really no positive empirical evidence that it did. This virus is never known to have existed in a lab, and the scientists who study these viruses as their professional specialties make their living, they do their work, they achieve their career successes by publishing what they find, finding dangerous viruses and announcing them to the world and warning the world about them. And in particular, now I'm alluding to, for instance, Zhengli Shi, Dr. Zhengli Shi, who runs the Wuhan Institute of Virology's coronavirus lab. She has made her living over the last 20 years by publishing discoveries of wild coronaviruses, analyzing them, analyzing the degree of their dangerousness. And if she had had this virus, logically, she would have published it. She might have gotten the cover of the journal Nature or the journal Science. There's no evidence that this ever existed in her lab or anything closer to it than that 96% similar virus that I mentioned, which she did find and announce to the world earlier. So 99-1 is a rough estimate of the odds.
Is there a connection between the destruction or preservation of habitat, the extinction or conservation of species, and the risk of diseases spilling over from the wild to people? Is there a connection in your own career, but also is there a connection in the wider world?
Yes, to say there's a connection is an understatement. It's all about that. It's all about human disruption of highly diverse ecosystems, bringing us in contact with the wild animals that carry an abundance and great variety of viruses. You know, the world is full of viruses. All of the animals and plants and fungi and bacteria and other microbes on this planet, cellular microbes, carry their own viruses. Viruses, of course, are not cellular. They're genetic parasites, but they have no cellular metabolism or anatomy. So they all carry viruses. And as we go into the great diverse ecosystems on this planet, cutting trees for timber, mining minerals, building timber camps filled with workers, building mining camps filled with workers who get hungry as they work and who need protein and who do not have any grocery stores in those timber camps in the Congo or elsewhere. They come in contact with wild animals. And when people capture wild animals live and ship them from one place to another to be sold live as food, that's another great opportunity for viruses, a situation of exposure, of close exposure of humans to the animals carrying these new viruses. And viruses don't have intentionality. They can't walk, they can't run, they can't swim, they can't fly. They ride in hosts and they then spill over accidentally into other hosts when opportunity presents. And because they are opportunists themselves, if they spill over into a creature and find that they can replicate themselves in this new creature because they are broadly adapted or very quickly adaptable, then they will do that. They will follow the Darwinian imperatives, the three big Darwinian imperatives. Make babies as much as possible, make copies of yourself, and distribute the copies of yourself in geographical space and extend yourself in time. Those are the Darwinian imperatives, and that's all viruses know how to do. And they will do that in a new host if they can, and spread. And if that new host happens to have 8 billion individuals on this planet, that the virus that spills into that new host will have won the sweepstakes of high Darwinian success. And that's this virus has won the sweepstakes because we're those 8 billion individuals.
Makes perfect sense. By the way, everybody out there, we very much welcome your questions. 1 or 2 questions have come in already. We won't lose them, don't worry. And I'll come around to those questions in just a moment. And I'm sure many of those will be on the latest book on Breathless. But before I turn over to those questions, 1 or 2 other quick questions from me. I've heard through the grapevine, David, that you're working on another book already.
I am finishing another book that will be published next May by National Geographic Books on conservation. It began as a collection of conservation stories that I had written for National Geographic magazine over the course of 20 years. They would send me, I was sort of their conservation beat guy, and they would send me to different countries in Africa or South America and other places and say, David, give us a story on Greg Carr and the Gorongosa National Park project. Give us a story on the organization African Parks. Give us a story on Chris and Doug Tompkins and their efforts to preserve wild landscape in Chile and Argentina and give it to people to make it public parks. Give us a story on, and that went on for 20 years, beginning with the point in winter of 1999 when an editor, a wonderful fellow at National Geographic, called me up one day, slight British accent, Oliver Payne, and I had never worked for National Geographic, and said, we're doing a big project on a fellow who's going to walk 2,000 miles across the Congo Basin to do a survey. He calls it a mega transect. His name is Mike Fay, and your friend Nick Nichols is going to be the photographer. We'd like you to be the writer. So that's where my relationship with National Geographic began, with Mike Fay and the mega transect. And those pieces are included in this book also. So this book is a pulled together. It's a new book in the sense that I've written a lot of new material, connective material, updates of all these conservation stories, a new forward, a new afterward, trying to put it in perspective. So it's essentially my treatise on, in my very humble opinion, although I've been privileged to have a lot of access and a lot of on the ground time, of what works in conservation and why it works, and why projects and efforts endure if they do.
So what does work in conservation, David?
Well, that's a huge question, particularly for this audience, because this audience knows so much. Some things that will be familiar to you. Community engagement works. You can't just draw a line around a beautiful patch of habitat in the middle of some jungle and say, we own this now. This is national park. Everybody stay out, because there are going to be local people who have lived there forever, who have needs and rights and expectations. So projects such as particularly Greg Carr's, the Gorongosa National Park project in Mozambique, is exemplary in that way. I spent time there, enough time there on the ground in and around Gorongosa National Park to see the incredible efforts that they have made to make that national park not just tolerable to the rural people living around it, but to make it the best thing that ever happened to the people in the villages living around it, in terms of help with their agronomy, in terms of help with their micro enterprise, in terms of girls' clubs for young girls to help keep them in school so they don't get married off at the age of 12. All of these things are being done there. And that's one of the things that's hugely important. Another thing that's hugely important is rigorous anti-poaching wardenship, and African Parks is exemplary on that. And there are things that Rainforest Trust does that are exemplary. Unfortunately, I wasn't well enough attuned to Rainforest Trust.
No apologies. First of all, I mean, you may or may not know that we support both, that our donors support through us both Gorongosa and Chinko in Central African Republic at one of Africa Parks projects, and we hope in due course to have other African Parks projects as well. So we share your enthusiasm for the superb work of both of those organizations.
And I knew as I was saying those things that there was a lot of overlap with the things that Rainforest Trust is already doing, and that the people who support Rainforest Trust also support some of these other projects.
Our donors through us are also assisting ANPN, the National Parks Authority in Gabon, that you were catalytic in helping to create, to expand Bateke National Park, and we hope in due course to help them with additional sacred sites for communities, and they have one or two more large areas that they would like to preserve, including near the border with Congo Brazzaville. So yeah, very completely enthusiastic about the organizations and people you're mentioning.
You're too kind to me. All I did was help to make Mike Fay and Lee White famous.
Absolutely. No, absolutely. Great. Well, thank you so much. I think we have a whole bunch of questions coming, and people, please excuse me if I don't necessarily take them in the order in which you presented them, but we'll try to get to most of the topics. And in general, I think I'll mention your name unless anyone wants me. If you want to remain anonymous, mention that in the question. But a question from our vice chair, and a great supporter of the effort to create national parks in Gabon and of African parks, and a great enthusiast about Gorongosa, Edith McBean, asks, do your scientific sources think there is another pandemic coming?
In a word, yes. Unfortunately, yes. The sources that I trust most and stay in touch with are still quite concerned about influenza, pandemic influenza, in particular, the possibility of avian influenza, high pathogen avian influenza, adapting well enough to humans to transmit readily from human to human. I mentioned Don Burke, I think. He was talking about that, again, 10 and 20 years ago. And scientists are still watching that carefully. And I'm working on an op-ed for the New York Times right now. I've been a little bit slow in finishing it because of the book publishing and my case of COVID, but it is on this subject. It's on the subject that, hey folks, don't forget about avian influenza. That could be the next big one. But that said, it's not inevitable that there will be a horrific new pandemic as bad as this one or worse, because besides knowing how capable these viruses are of crossing into humans and adapting for transmission human to human, we also have learned a lot about how to watch for them. We need much more viral discovery and surveillance. We need to be watching so that when the next really dangerous virus spills over into a group of unlucky humans somewhere in the Four Corners area of the US or in an African village or in a suburb in Australia, that it is contained before it moves from being an outbreak to being an epidemic, and from being an epidemic to being a pandemic. And that's possible with surveillance and response. But the surveillance and response we have now is not good enough. We need much more attention to that, much more resources, and much more cooperation devoted to outbreak surveillance and response, early warning, capture, contain before the thing spreads.
Thanks. A longtime supporter of Rainforest Trust, Kathy Donahue, has two or three questions. She says, I think Marburg was accidentally released from an Austrian lab. How was that escape release first verified? And then she also asks, why do viruses spread? What viruses do we spread to other species?
Okay. Well, first of all, Marburg. Yeah, it wasn't an Austrian lab. It's Marburg, Germany. African monkeys were shipped up for medical research in 1967. They were captured in the Lake Victoria area. I think all of them, or at least most of them, were captured on an island in Lake Victoria. They were of a species that I think used to be called African green monkeys, and maybe now they're called one of the guenons, but I don't know that precisely. Anyway, they were shipped up for medical research, and they were infected with this terrible virus, Marburg. It's a filovirus, same family of viruses as Ebola. And they made people sick. So it's not what people think of as a classic lab leak of a virus. It was a leak from monkeys, not from test tubes. There were handlers and scientists who were coming in contact with these monkeys that had just been flown up from Africa. And the monkeys were carrying this virus and spread it to people. And it infected a couple of dozen people, and it killed a high percentage of those. And this was the very first recognition of Marburg virus. It was 1967, so it was before Ebola had even been recognized. It was this new virus that was filamentous. Scientists isolated it, characterized it, gave it its own family, and gave it a name, named it after the city where it broke out, which you don't do anymore. But that's why it's called Marburg virus. In terms of the viruses that we pass to other animals, well, the real danger is respiratory viruses from us to apes. So people involved in, for instance, ecotourism to see the mountain gorillas in Rwanda, they are obliged to be very, very careful that visitors who go up to spend time with the habituated groups of mountain gorillas are not carrying respiratory viruses, are not carrying flu or common colds or anything like that. They don't sound like I sound right now. If they do, then you don't go on the day trip up to see the gorillas. Protecting them. There are cases of other viruses, I think, including polio, suspected as having gotten into the chimps of Gombe. Jane Goodall talks about this in one of her books. Early on in her research project, there were some chimpanzees at Gombe stream that were dying of what seemed to be polio, and it was suspected that they had caught it from people in the village that services the Gombe Reserve. But respiratory viruses in particular, the risk of them getting into great apes in particular, because they're so close to us. And then there are also, it's very common for us to pass viruses to other animals. For instance, SARS-CoV-2 now infects something like more than 40% of the white-tailed deer in Iowa test positive for COVID, for SARS-CoV-2. Mink all over the European landscape have acquired this because mink farming particularly in the Netherlands and Denmark was done at high intensity for fur, raising American mink for their fur. And there were animal handlers and workers who went in there and they were positive for COVID, and they infected the mink. And a certain number of mink each year escape from mink farms. They go wild, and the American mink has already gone feral on the European landscape in the Netherlands and Denmark, among other places. And so escapee minks from these farms where people who have been infected with human adapted coronavirus are now at large on the landscape of Europe.
I can't, of course, look into Kathy's mind behind the question, but because I know that many of our core supporters believe very strongly that other species deserve a place on the earth as much as we do, I'm expecting that her pointing out that we're as much of a risk or more of a risk to other species, even around disease, than they are to us, may have a moral dimension to that question as well as a scientific dimension.
Absolutely. And by the way, the good news on that story is that commercial mink farming in the Netherlands and Denmark, if my information is correct, it's dead, it's gone, it's finished. They were phasing it out. But when this happened, they said, that's it, done. But of course, we still have 35 billion chickens in captivity, and that's related to the threats of avian influenza.
We have some detailed questions from both trustees and council members. I'll go to the longest one. Our former board member, and in fact, the architect of our brand, the person who is responsible for choosing and developing our logo, as well as much of who we are, Jeff Zach, who recently rotated off the board, and we're missing him, but happily he stayed on our council. He has written, "I'm a little breathless here listening and look forward to reading your book." He's a bit of a fan of yours, you may remember. A question: what lessons have scientists learned from COVID-19? What about gain of function research? Even though this virus likely did not come from a lab, are the obvious risks from intentionally making viruses more infectious still seen as being outweighed by the benefits?
Yeah, that's a very tangled question. Question that,
do you want me to untangle, or have you got it?
No,
even through the COVID blog.
Actually, the question is crystal clear, but the subject is tangled. I could I could spend my next year just working on that subject and writing something about gain of function, but I talk about it in the book. I talk about this question, about the values and the risks of so-called gain of function research, and I can just barely scratch at that a little bit here. First of all, on the face of it, it sounds to some people reckless. You take a virus in the lab, a virus that you've been able to culture, because it's important to remember the distinction that between a virus that has been grown live in a lab and the genome of a virus that has been sequenced from a sample. The genome is not a live virus. The genome is like the script of the play Hamlet, Prince of Denmark, but it's not the performance. The live virus, if you manage to grow it, is the performance. And live viruses are very difficult to grow in the laboratory, particularly from samples such as bat poop. So there are constantly efforts to grow live viruses that don't succeed, but it's easier to sequence a virus. So you have the sequence and you can study it. But in some instances, you do have a live virus and you wonder, well, this does not seem to be transmissible from human to human. If this were transmissible to human, what sort of adaptations, what sort of mutations would allow it to gain that capacity? So you experiment with it. You might experiment by putting it into ferrets, which in terms of respiratory infections have a physiology very similar to humans. So ferrets are the model animals of choice for this. Maybe it's an avian influenza, and you wonder, what would it take for this avian influenza to become transmissible among humans? Well, let's mess around with ferrets and see if it evolves to become transmissible through the air between ferrets. Oh, it has. Okay, let's look at those mutations. Well, how does it differ from the other virus? What has it acquired? Now we have a search image for what are dangerous steps on the path toward a transmissible avian influenza in humans. But from the other side, people say, well, you've created an avian influenza that's now transmissible among ferrets and therefore among humans. That's crazy. That's dangerous. Neither of those points of view is idiotic. But I've just spent much of the last 2 weeks talking with some of the best avian influenza researchers in the world. And one of the things they were telling me is these gain of function experiments are crucial. They are extremely valuable to what we're doing, trying to understand how avian influenza might become much more dangerous than it is in order for us to know what to watch for. so that's sort of the first level of response to that. But that's a long conversation, and an important conversation.
Jeff works in the insurance industry. And so I expect he's particularly attuned to unexpected consequences and underestimates of small probabilities. But
by the way,
as a follow-up question to Jeff's, go ahead.
One other thing about that.
Yeah.
I talk more in the book about the particular laboratory work that was done with coronaviruses, certain coronaviruses from bats, to see how they might change and become more adaptable. And it's a very complicated story, and I won't try and recreate what I say in the book. But what some people see as a gain of function experiment is what other people see as something that has not created a creature new to the world and therefore a dangerous monster. They see it as something as work that has created something that already exists in the wild, but we can't study it in the wild. And now we have a version of it in the laboratory, and we can study it in the laboratory. And that's why there was that very conspicuous interaction between Tony Fauci and Rand Paul when Dr. Fauci was testifying before the Senate. And Rand Paul was saying, how dare you support this gain of function research? And they were talking about a particular scientific paper about coronaviruses. And Tony Fauci was saying, Senator Paul, you don't know what I'm not going to do the Brooklyn accent. I'm too respectful for that. But Senator Paul, you don't know what you're talking about. Let me make that official. You don't know what you're talking about. That was a question of what is or is not gain of function.
So super complicated. From another council member, Callie Broadus, who is I'll get the exact title wrong, but an explorer in residence of some kind at National Geographic. So shares your ties to that wonderful organization. She writes, how do you differentiate between local and indigenous communities who have historically lived among and consumed wild species, and the example of mining camps entering similar wild spaces and potentially unearthing zoonotic diseases?
Well, mining camps, mining workers entering wild places and potentially coming in contact with dangerous viruses, acquiring zoonotic diseases. That is an important category to think about, but it doesn't even abut the category of concern for local people that I'm talking about. Although there is some area of ambiguity there, and I'll circle back to that. But for instance, in the Eastern Democratic Republic of the Congo, there are mining camps to extract coltan, this strategic mineral coltan, from the earth. And coltan is important because it's in these, it's in our laptops, it's in our smartphones in the form of tantalum capacitors. Can only be mined a few places around the world, but it's absolutely essential to these fancy consumer electronics. And the people who mine it, men and women, are out there digging in the dirt, living in camps. There are no grocery stores. Their employers are not supplying them with, high quality domesticated protein. They are in a situation where most of them probably, to have any protein, have to eat bushmeat, have to eat wild animals. So they're eating porcupines, they're eating rats, they may be eating bats. And they're exposing themselves to new viruses, zoonotic diseases. And that's why we should remember that we all own a piece of responsibility for this problem, that it's not just people in China who want to have a live porcupine butchered for them to take home for a family feast. It's all of us doing things that have impact on the natural world in ways that bring humans and animals closer together so that viruses can pass between them. The coltan example is the best I know of to remind everybody that this is partly your responsibility. But there is a distinction, this is back to the ambivalence thing, or ambiguity. The people who have lived on a landscape in Papua New Guinea for 40,000 years, making their living from wild resources, to me have a different level of legitimate claim than, for instance, my dear friends, the cattle farmers of Montana, who will be very proud if they can say that they have lived in Montana for 5 generations, making their living ranching cattle on the landscape. There is a difference there. The people who are raising cattle on the landscape deserve respect, deserve being listened to. They are stakeholders, but they are not indigenous people. The Crow and the Blackfeet and the Assiniboine and the Northern Cheyenne are the indigenous people. But everybody comes from somewhere. Everybody has come from somewhere. And even the Crow and the Blackfeet came here from somewhere else 10,000, 12,000 years ago. So there are differences, there are gradations, and it requires sensitivity but also rigorous thinking to figure out how to adjudicate those claims.
So back when you were talking about what works in conservation, and you spoke about how central it was to engage with Indigenous people and other people who've been living in a place and have legitimate claims to that place. The examples you gave of your favorite successful conservation organizations were organizations which indeed engage with local people, but are not Indigenous led. And today, we do have many people from the Indigenous communities in North America, South America, elsewhere, suggesting that much more of our conservation efforts should be in supporting Indigenous people in their direct stewardship of land, as opposed to creating traditional national parks. What's your view on that healthy and complex debate?
Well, one thing I can say about that is that exactly what you just described, James, is why I'm glad and encouraged when I look at the Rainforest Trust portfolio of projects and see that you all have been finding small Indigenous organizations, little startups to partner with in these efforts. And I think that's a hugely important and correct approach to take. It's not that necessarily that the local organizations are going to replace the international donors in the international organization, but that they can work together, and it's just a crucial form of partnership. And you know far better than I do the list of those local partners that you've been developing relationships with, and I'm certainly glad to see that and hope that we can do more of that.
Yeah, I mean, we're very excited to have you on the board for many, many reasons, not least that we've never had so many people come to a webinar before, but also because you kind of promised us if you joined the board, you would be a bit of a moral conscience and ask again and again what our projects were doing to benefit local and Indigenous people in the area, and also what we're doing to increase the diversity of our staff and council and board. So that's very welcome to have you play that role among the many others that you will play.
Moral conscience is kind of a glorious phrase that I cringe from, but I certainly, you're right that I said local people. How are local people involved? Are local people being taken into consideration in each of these projects? And it's not just, it is a matter of morality, but it's not just a matter of morality too. It's a matter of practicality, because without that sort of engagement of the local people, projects don't last. They're not destined to last for generations. In order to make these projects work, one has to make sure that these activities are the best things that ever happened to the local people.
Two more questions from people who've joined us today. One from another long-term supporter, Frank Smith. And Frank writes, this is a question near the end of the talk. Okay, well, we're near the end of the talk. Have you thought of writing a follow-up to the Song of the Dodo, a book I think of as very relevant to the work of Rainforest Trust? much of the science has changed, and you are probably very well placed to tell us what is new about island biogeography and related matters. And I would just put in that, of course, some of the key people in Song of the Dodo are connected to the Rainforest Trust, including Ed Wilson, who passed away last Christmas and who was on our board, but of course, who was the co-author of the theory of island biogeography, and then later its application first to reserves, and his colleague Tom Lovejoy, who applied it to the design of reserves in an experimental way, and also died within a day of Ed. And then both of them really pushing for expanding protected areas now with Ed's vision of half Earth. Yeah. But back, does Song of the Dodo need a sequel?
Well, with all due respect to Frank, I would say the details have changed, but the principles have not changed. And that's a book, a long book that combines a lot of detail, a lot of case studies, a lot of reporting on the ground, a lot of crazy wild animal stories and portraits of fabulous habitats. And the circumstances of those places have changed in many cases for the worse, but the principles haven't changed. We still know that you need big areas if you want to preserve richly diverse ecosystems, but that consideration is complicated by the wisdom of someone like Dan Simberloff, who says, yeah, but sometimes allocating your conservation resources to small places allows you to preserve more very particularized endemic situations than you would if you just spent all of your money on large-scale connectivity. Those conversations are still going on, are still informing conservation biology and conservation activism. But the principles haven't changed, as I see it.
I'm going to squeeze in one last question from our esteemed board member joining us from Scotland. So thank you for staying up. Kim Stewart, who writes literally, and I'll let you interpret, can we do it? Are you optimistic? Where do you find hope?
Tough, good question, Kim. Thank you. I am not a Pollyanna. I am not a naturally optimistic person. I am a diagnoser of problems in a lot of the work that I do, but I also believe that that hope is not a mood. Hope is an act of the will. Hope is a resolution, and we absolutely are required to keep hoping that we can do it in order to energize us to keep doing it. There is nothing more important on this planet, I think, and you all think too, than trying to prevent the total loss, the catastrophic drawdown of the wonders of biological diversity on this planet. And it's a rearguard action. It's pushing a huge boulder uphill. There are heartbreaking losses every day, but then there are little victories. And we all know the old adage, in conservation, all of your victories are temporary. It's only your defeats that are permanent. We can't think that way. We have to have hope. And that's why people like Ed Wilson, Tom Lovejoy, two good friends of mine, both of whom sort of drew me into that 8-year effort to write the Song of the Dodo, or at least endorsed it as I went in and said, go, yeah, you should do this. You should do this. And our friend Jane Goodall, those three people were and continue to be preciously valuable to this planet, not just because of what they've done or what they've understood or what they've said, but because they were tireless and inexhaustible in their ability to hope and to share hope, to convey hope. So yes, I'm hopeful, because if I weren't making the active will to be hopeful, I would be I would have no reason to get out of bed in the morning. I would be sitting here at 2:30 in the afternoon in Bozeman, Montana, drinking cheap wine out of a paper bag.
I mean, I think you've taken that question and turned it around back to all of us who are here with you today and told us that we have no less than a moral obligation to be hopeful.
Yes.
Because that's the only way that we will all together achieve what we need to achieve in saving biodiversity and stabilizing our climate and leaving a decent world to both our own descendants and the descendants of all the other plants and animals on Earth. So thank you.
What he said.
It is such a pleasure to talk with you. I'm scared to think of how razor sharp you would be in the absence of the COVID brain fog. It's terribly kind of you to have joined us in spite of being a little bit ill. And we just can't wait to work with you over the next 9 years on the Board of Rainforest Trust.
Great pleasure to me too, James. And thank you all for tuning in and listening to this. And I really, really look forward to working with Rainforest Trust. It's a great privilege to be invited to be involved.
Thanks everyone for joining us, and be in touch. We all have telephones so we can continue the conversation.